Before the discovery of the molecular structure of deoxyribonucleic acid, more commonly known as DNA, there was not much known to the scientific community about its true potential as a molecule. In the 1940s, tests were done by British biologists that proved DNA to carry the instructions for the function and operation of organisms. What mysteries of life lay in its structure? With the world's top scientists working for a solution, it was only a matter of time. Due to this intense competition, the world would soon know the names of two little-known biologists with a passion and a photograph. In the early 1950s, Rosalind Franklin, an X-ray crystallographer at King's College in London, unwittingly made the partial discovery of the molecular structure of DNA. Unfortunately, her incomplete data was misappropriated, completed, and published by two scientists, James Watson and Francis Crick, with the illicit cooperation of Franklin's partner, Maurice Wilkins. This put an end to a 3-year race to discern the DNA structure. The discovery of the molecular structure of DNA rocked the scientific world. It was and continues to be a turning point in history because it revolutionized the fields of science, medicine, agriculture, forensics, and more, as it gave us the unique blueprint for life as we know it.
Nucleic acid was discovered by an unnamed chemist in 1868, 2 years after Gregor Mendel's groundbreaking discovery of nuclein that earned him the title of the father of genetics. It was later found that Mendel's nuclein comes in two forms, deoxyribose and ribose, later named deoxyribonucleic acid or DNA and ribonucleic acid or RNA. Until the 1940s, scientists assumed that DNA had something to do with genetics, but they still thought it was too simple and insignificant to carry the complicated instructions necessary for building an organism. James Watson was an American biologist who secured a position at the Cavendish Laboratory at Cambridge University, England in 1949. His director was the renowned crystallographer and biologist Sir Lawrence Bragg. X-ray crystallography is the science of taking a photo of a molecule using X-ray beams, crystals, and photoplates to create reflections of the target molecule. At the lab, Watson became fast friends with Francis Crick, a biologist and crystallographer at the Cavendish Laboratory with an exuberant personality. After meeting, the pair discovered that they shared the burning theory that DNA was the genetic material.
They were not alone in their quest for the DNA structure. 55 miles away at King's College, London, two other scientists, Rosalind Franklin and Maurice Wilkins, were working on the same problem. Wilkins was an old friend of Crick's and had been the head of King's crystallography department until Franklin, a world-renowned crystallographer and a vigorous researcher, was hired in 1950. While Franklin was researching, Watson and Crick were speculating that DNA was a helix or spiral in shape and building conceptual models, as was Watson's hero, the famed Caltech biologist Linus Pauling. Pauling was arguably the greatest biologist of the time, and yet another competitor in the race for the structure.
Because of Franklin's superior knowledge of crystallography, Crick sent Watson to one of her lectures to attempt to gain insight into her approach. At the lecture, Franklin talked about two forms of DNA, the dry A form and the wet B form. The slides that Franklin showed helped fuel Watson and Crick's theory that DNA was a helix. As Watson had neglected to take notes during the lecture, he misinformed Crick about the correct number of water molecules surrounding the B form. Watson's mistake caused Crick to misinterpret the data for the model they subsequently built. After showing their inaccurate model to the King's team in 1952, Watson and Crick were forced to listen to Franklin ridicule their work, which was a triple helix. She went into great detail explaining how their model did not follow her findings, which were indeed correct. An opportunity then presented itself. Linus Pauling released to the public that he had discovered DNA structure. Watson and Crick feared the famed biologist had actually done it until they examined his data and found that he had made the same mistakes they had made on their first model. They worked in earnest over the next few weeks, knowing that Franklin was preparing to leave King's for another position. Before her departure, Wilkins contacted Crick, saying that he had some of Franklin's photos that he was willing to share with the Cambridge team. Crick sent Watson yet again to collect information, this time with explicit instructions to pay attention, as seeing how well her past findings had turned out.
Watson took great interest in a photo of the wet B form of DNA, labeled as Photo 51. He had gleaned enough information from Crick's ramblings on crystallography to realize that the photo was solid proof that the structure of DNA was a double helix, the very theoretical structure Watson and Crick had conceptualized. Franklin must have taken it without realizing exactly what it was. Using the photo's information, Crick and Watson were able to come up with correct measurements and construct a second, accurate model. The structure Watson and Crick had created was a helical ladder that could separate into two halves like a zipper. Watson and Crick's findings were published in August of that year, hailing the two biologists as the discoverers, never mentioning that Franklin had actually done it.
Suddenly to see the molecule which is responsible for heredity and which makes possible human existence was a very big step in such a man's understanding of himself, in the same sense that Darwin really knew that human species, that we weren't fixed, that we were changing, and that was bound to affect your attitude to everything.
Watson, Crick, and Wilkins were awarded the Nobel Prize for their work in 1962. Franklin died of ovarian cancer at age 37 in 1958 and was not given credit for her Photo 51 that was the catalyst to the DNA discovery. She never realized how her role in the discovery had completely changed the whole scientific view of life. Following this discovery, as an after effect, the Human Genome Project, a project to sequence and catalog the whole human genetic code, was created.
The after effects of the discovery of the molecular structure of DNA are numerous, affect many scientific fields, and have many practical uses. For instance, the medical world is using the information of the DNA code and structure to discover new cures for diseases and conditions by locating the proper genetic switch. Also, this technology could eventually be used to determine the traits of a child in utero, which would likely cause an ethical debate. Another application for this knowledge is cloning. Scientists are able to clone animals, the most famous example being Dolly the sheep, who in 1997 was created using DNA from a donor sheep and was an exact genetic replica. It is not yet possible to clone humans, as our DNA is the longest and most complex in all of the animal kingdom. Nevertheless, when and if it does become possible to clone humans, one can expect that there will be intense ethical debate. Stephen Rice, professor of microbiology at the University of Minnesota, reflects on the after effects of this discovery and why it was a significant event for the scientific community.
In my own opinion, the biggest turning point in the history of biology was the discovery of DNA, because that transformed biology from biologists thinking about organisms to actually being able to think about the genes of organisms and how genes worked.
Another field that can use the revolutionary discovery of the genetic code and structure to its advantage is agriculture. Scientists can utilize data on the genetic code of a crop to make modifications that could save lives. For instance, wheat could be more climate and disease resistant, while a variety of tomato could be modified to yield larger, more plentiful fruit. Today, DNA is also used extensively in the area of forensic science. DNA from a crime scene such as blood, hair, or saliva can be utilized to catch a criminal. Federal laws have been passed requiring all jails nationwide to submit samples from criminals to a national DNA database that can help clear the names of the innocent or confirm the guilt of the prosecuted.
The discovery of the molecular structure of DNA has completely revolutionized and bettered our lives by giving us as a scientific community, nation, and world the blueprint for life, the key to understanding ourselves as a species, as a people, which may be the final step to truly knowing who we are.